never put that in
Eukaryotes
the
have
exam.
organelles.
Which one?
Eukaryotes.
Eukaryotes have organelles and
Prokaryotes.
prokaryotes do not have. Okay? Give me an example of organelle. Mitochondria, chloroplast, Golgi apparatus, endoplasmic reticulum, all those. Okay? What are the difference? Yes.
The size of
Go
the
on.
petitoplaken.
The what?
The thickness and the thin petitoplaken.
Thickness of
Yeah, of the cell wall.
cell wall or
petito-
peptidoglycan. You
yeah.
say peptidoglycan.
Yeah.
So-
You have thick petitoglycan.
Which one?
Eukaryote has thick petitoglycan and prokaryote have thin.
So I have how many students here? More than 14. And you are there, no reaction. Did you hear that? Can you repeat what you said?
I said the petitoglycan, the eukaryote have a thick petitoglycan and prokaryote have thin petitoglycan.
Rara, you agree with that? Okay, what do you think?
I think you might have confused the gram staining.
Yeah,
The difference with the bacteria and the cell wall.
within the bacteria, do you
Yeah.
have peptidoglycan in eukaryotes?
No.
And all the eukaryotes don't have cell wall. You and me, we are eukaryote cells. You don't have cell wall, okay? So the one like plant, like fungi that have cell wall, the cell wall is made of another chemical, another sugar than the peptidoglycan, okay? In plant, the cell wall is made of what? Cellulose, okay? So what are the differences? The size. The prokaryotes are very small. Remember in the lab how we have hard time to focus them. But if you have eukaryote under microscope, it's easy to see. Okay, because they are larger. So this chapter, chapter 12, is about classification. Okay? Classification in each group. You see what do you use to classify the members in that group. Here we have three kingdoms. Bacteria or prokaryote are part of Monera, kingdom Monera. Here we have kingdom fungi, kingdom protists that comprise algae and protozoa, and kingdom animalia, where you have helminth, you will have also arthropods, okay? So those three kingdoms, we are going to study them here, and each time we will determine what criteria we use to classify the members of the kingdom fungi, of the kingdom protists, and so on, okay? So when you study, you go like that to from overall to the small details. Let's start with the kingdom fungi. The fungi are a group that has its own characteristics that distinguish it from other living organisms. So the fungi are mainly chemoheterotroph. What that means? They do what?
They make their own food.
Who make its own food is autotroph, right? Chemoheterotroph, they get their energy from, not from light, but from chemical reaction. We are chemoheterotroph. Another good characteristic of fungi here, their shape or morphology. They have both. They have unicellular, only one cell. Those are what we call yeast. And you have the mold that are multicellular. So among the fungi, you can have the members that are made of one cell, or many cells. And also, the fungi have two types of reproduction. They have sexual reproduction and asexual reproduction. What is the difference between sexual reproduction and asexual reproduction?
The number of parents.
The what?
Number of parents.
Number of?
Parents.
Okay, of mate, right?
Yeah.
Oh, okay.
Yeah.
So, in sexual reproduction, you have two. So the baggage, the genetic baggage that the offspring has will come from two parents. But in asexual reproduction, you go from one parent that will break down through binary fission or fragmentation or schizogony. You have all kinds of asexual reproduction, okay? But the fungi, they perform both, sexual and asexual. How do fungi reproduce? Come on, my student.
What's up?
How do they reproduce,
Which ones?
the fungi?
They bud.
They do what?
Budding?
Budding, yeah, that is a possibility, yeah. Fragmentation is another. Spore. The fungi, when you have the spores that are carried by wind or water or bird, where the spore land, if the soil is good and is wet, is moist, then the spore will germinate, okay? So in fungi, the spore is a means of reproduction. In bacteria, what do you know about the spore in bacteria? Yeah. The what?
It's a means of survival.
Yes. Do you have reproduction during endospore? No. So you see in bacteria, it's just a dormant state waiting for better condition. No reproduction because you don't have enough metabolism for reproduction. But in fungi, the spores are for reproduction. So you can be asked to compare and contrast the role of spore in bacteria and in fungi. That makes sense? Okay. So since the spore is means of reproduction, the fungi will use type of spore for the sexual reproduction and another type of spore for asexual reproduction. So they have two types of spores. You have sexual spore and asexual spore to use in their reproduction. The study of fungi is morphology. Now we did say that, I told that classification. For the fungi, in this chapter, for this class, we will use two criteria to classify the fungi. One is the shape or morphology, and the two will be the sexual spore. Let's talk about the morphology or shape in fungi. Based on the shape in fungi, we can have three groups. The classification, we can classify fungi in three groups. The group that is called mold. Mold are made of type of filaments. Those filaments are called hyphae. When you have a lot of those filaments that can break fragmentation to give a new and so on, when you have pack of them, that is called mycelium. So mycelium is a pack of hyphae. Okay? So the two words here are part of vocabulary. Okay? The second group in shape is yeast. Yeast are unicellular. Those are made of cells, not filament. cells and one cell at a time. So the yeast are unicellular. They reproduce through division. You have two types of yeast based on how they reproduce. Some will divide symmetrically and give two offspring that are identical in size. So that division is symmetrical. You call that type of yeast the fission yeast. Fission yeast divides symmetrically. Example. In this chapter, the example that I will be giving you for the exam, since you have the terms that are a little bit not everyday terms, so I will emphasize on the genus, except a few that I will tell you that you have to know the species, okay? So example of fission, fungus you have Saccharomyces cerevisiae. So Saccharomyces is an yeast that we use, we call baking yeast. We use it in the kitchen for bread. We use it for fermentation, okay? So it's Saccharomyces. And you have the yeast that are budding yeast. How do they divide? Asymmetrically. That means the parents, on the parents you have bud that will develop, and then that bud will detach itself, become a new yeast, and so on, okay? And the budding yeast, a good example is Candida albicans. Put asterisk because for Candida albicans, you have to know the species too, Candida albicans. Candida albicans will give, this is the parent, and the bud will develop, and then detach itself, become a new yeast. It happens sometimes, especially in the case of Candida albicans, that the bud fails to detach itself from the parents, and then at its turn, you have the bud that forms. And if it doesn't detach itself, so you can have string of buds, okay? So the string of buds will look like hyphae, like a filament, long, like a filament. That's why it's called pseudo hyphae. Vocabulary, very important. Why? Because the fungi like Candida albicans need to be in this structure, pseudo hyphae, to be pathogenic. If it's not in that structure, it's less pathogenic. So that is the structure that when Candida albicans is in that structure, can enter deep in the tissue and continue to cause the disease. Okay? The third shape, we are classifying based on the shape. The third is dimorphic. Di means two. So dimorphic are the fungi that freely can switch from mold to yeast, back and forth, depending on some factors in the environment. I give you example. In the lab, if we take a plate with the media for fungi, where we grow the fungi, that is called saburo. So if you take two plates, you streak the Candida albicans, you take one plate, you put in the incubator, the second plate you leave on the shelf at room temperature. When you come back after, you will find that the one in the incubator where you have 37 degrees Celsius, if we take some of that, of the colony that grow, you put on the slide with a small drop of water, mix and look under the microscope, you will have the circle, the cells. That means the candida albicans at 37 degrees Celsius is east, okay? But you take the one at room temperature, you fix it the same way, you look under the microscope, you will see the filament. So candida albicans at 37 degrees Celsius is an yeast, that means it's made of cell, and at 25 degrees Celsius is a mold made of filament, of hyphae, okay? And you will find that most of pathogenic fungi are in dimorphism because they are free to cause disease, all the situation are good for them, okay? So mainly the pathogenic ones are dimorphic, okay? So if we classify based on the shape, the fungi, we will have mold, yeast, and dimorphic. Are we together? Any question? The hyphae that form mold, there are two types of hyphae. You have septate, that means you have division in the cell, that those divisions are called the septum. So you have the division. Although you have divisions, the nuclei can travel freely through the hyphae. Those are called the septate, septate hyphae because of septum. While this type, where you don't have division, you don't have separation, you have choanocytic hyphae. So you have to know what is choanocytic hyphae, what is septate hyphae, and be able to recognize which one is which.
You said those four molds?
The what?
You said those two four?
Hyphae. Hyphae form this form mold. So let's see the tree shape. You see the small line here. This picture is not the best one. You have the small lines here are the hyphae. But what you see here at the end of hyphae, you have a ball, the black ball here. You call it sporangium because that contains the spore, okay? So when those spores are mature, when the sporangium is mature, you will have the crack to free the spore. And now the spore dispersion can be secured by bird, butterfly, or if it's near the water or the wind. So all those are good for the dispersion of the spore. And wherever the spore lands, you have germination, like we see here. The spore will germinate if the conditions are right, will germinate and give a small hyphae that will grow out of the spore, and you have adult hyphae, and the cycle will continue. Example of mold, please put down. Example of mold, you have Aspergillus. What do you know about Aspergillus? Not necessarily this species, but Aspergillus. Did you ever heard about Aspergillus? No? Who say yes? Yes. Go to chapter 1. Go back. Aspergillus is a fungus. Here is a mold that can produce a chemical. The chemical that is carcinogenic. And this chemical, mold infect the peanuts. And on the peanut can produce a chemical that is carcinogenic. What is that chemical? Aflatoxin. Okay? Do you remember that? And mycotoxin. Okay, let's go to yeast now. The yeast, the budding yeast, you see the yeast here. You have the bud that will form, and then will detach itself, become another yeast. We see the detachment here. You have a new yeast. Okay? Here, the bud is already detached. You see the remaining is the scar. So sometimes if you look under the microscope, you look at the yeast, population of yeast, you can see some of them with the scar. And some have many, like here we have 1, 2, 3, if I've counted the other side. So the number of scar on one yeast can give you an idea about age of that yeast. If it's an old yeast that have many scars, or is a young yeast. Okay. Remember, example, we have Candida albicans. The third is dimorphic, switching between yeast, mold, and back and forth depending on the temperature. When it's 37, you have yeast, like growth. When it's 25, you have mold, like growth. An example of dimorphic to facilitate, you can use again Candida albicans. This is to tell you that yeast is very, very important. We used to call Candida albicans yeast all the time without thinking that can be molded too. Why? Because our body is 37 degrees. So when we are infected with Candida albicans, when we have problems with Candida albicans, we talk about the yeast that is growing on 37 degrees. Okay? That Candida albicans can be also molded if you put it in 25 degrees Celsius. We know that the yeast infection is caused by the Candida albicans. So now that is the first type of classification of fungi based on the shape. Now we did say that they have sexual reproduction and asexual reproduction, that will use two types of spore. For the asexual spore, I will just show you, we get to go through, because you cannot use asexual spore for classification, because many fungi have the same type of asexual spore. So what is used here for classification will be the sexual spore, okay? So here we have example of asexual spore for asexual reproduction. Here we have asexual spore. You see the small shorty that are straight, yellow, arthro because they are straight. Those are the asexual spore. You have pseudo-hyphae. The branching is also asexual spore. You have this type of chlamydoconidium, like worms, asexual spore. But you don't use asexual spore for classification. This one is also asexual spore, okay? Let's talk about sexual spore, because that's what we use for classification. Before we talk about sexual spore, let's talk about the reproduction in fungi. The reproduction, that is sexual reproduction in fungi, has 3 phases. Very, very important. The 3 phases of sexual reproduction in fungi. You have plasmogamy, which is fusion of cytoplasm. You have 2 haploids that means N chromosome, that will act one as plus, the other as negative. The plus will penetrate its nucleus in the recipient. So what is the result? You have one single cell, one single cytoplasm, and inside you have two nuclei. Okay, so plasmogamy is fission of cytoplasm. Then that will be followed by fission of nuclei. So karyos is nucleus, karyogamy. Now in that one cell with two nuclei, the two nuclei now will fuse. Okay? So you will have at the end one cell and one nucleus. But that nucleus will be diploid, two chromosomes, because that came from one haploid and one haploid to have two N chromosomes. I guess we understand each other when we talk about haploid, diploid. Do we?
Yeah, Professor. So, I just want to get the clarity of the first one.
Answer to my question first and then give me a question after. I'm asking if we understand what is diploid, haploid. Okay. Yes.
Yeah, they said the first one is the fusion of the cytoplasm. So is the
Is what?
I said I want to get the, can you help me with the clarity of the first one?
The plasmogamy.
Yeah.
Okay, you have two cells that come together. Those cells are haploid. That means that their nucleus, there's only one type of chromosome. Like us, that will be 23 chromosome, while diploid is 46. Okay? So the two cells will fuse, the two haploids will fuse the cytoplasm, but their nuclei will not. They will stay. So you will have a cell that has one cytoplasm and two nuclei. Those two nuclei are haploid each. Then you go to karyogamy, where the two nuclei will fuse to form a diploid. And now once you have a diploid, the cell have to undergo what is called reductional division, what we call meiosis. We go from 2N to N. We go from diploid to haploid. At this point, at the end of meiosis, we have two haploids that will now undergo the plasmogamy. So you have the three here phases that keep going one after another. Now, what are the sexual, you have a question? Okay, so what are the sexual spores in fungi? In fungi, you have three types of spore. You have zygospore. Zygote means egg after fertilization. So zygospore are the spores that result from fission of two haploids. And you end up with zygospore. Then you have ascospore. This is the type of spore that are protected. They are formed inside of sac that protect them. So they are hidden inside of the sac, and that sac is called ascus. So ascospore are inside of ascus. Then you have basidiospore that are not protected, are not hidden. They are external spore. They stick out like club. They stick out. Okay, those are the three types of sexual spore that fungi can have. From the three type of sexual spore here will correspond three groups of fungi. Okay, so if we want to classify the fungi based on sexual spore, we will take the fungi that carry the zygospore. They are called zygomycota. Mycota means fungi. So zygomycota carry the zygospore. What do you want to retain for zygomycota? This is scientific name zygomycota. In some books, you'll find Zagomyces. Because they were, those are the Latin terminology. They were calling it Zagomyces before, and then they changed myces by mycota. So both are right. The common name of Zagomycota is conjugation fungi. Conjugation because you have two haploids that came together, you have fusion, and that process conjugation. So Zagomycota or conjugation fungi. Example of Zagomycota, you choose one of those. Like Rhizopus is the mold that grows on the bread. Okay, Mycor is more pathogenic that cause systemic mycosis. So for the example, I will leave to you. When you study, you choose one. Okay? You choose one in each category, the one that you want to pretend. Then you have the second group that carry ascospore, you have ascomycota. Since the ascospore are protected in the sac that is called ascus, the common name is sac fungi. An example of ascomycota, you have all those examples here. You will choose one that you want to use as example for ascomycota. And finally, you have basidiomycota. Since the basidiospores stick out, they are external, they stick as club. This group, the common name is club fungi. Example, you have Cryptococcus neoformans. To not confuse with bacteria because of coccus. This is an yeast, Cryptococcus neoformans. You already heard from some university or colleges that they found a few cases of meningitis. Like a few years ago, it was Tyson University, few cases, very sporadic cases of meningitis, and then they found some in Virginia and so on. Those meningitis is inflammation of the meninges, the membrane around the brain. So this can be caused by the fungus yeast, Cryptococcus neoformans. So they will talk about fungal meningitis. Or that can be caused by a bacteria like Neisseria meningitis, and they will talk about bacterial meningitis. So you want classification based on sexual spores. If you are asked to give classification of fungi based on sexual spore, here the classification is fungi, it's not spore. So you have to say Zagomycota, Ascomycota, Basidiomycota. Don't give Zagospore because you are asked about the fungi. You be careful. For a long time, the fungi were not well studied. The research was more on the bacteria because they thought that fungi are not important in our life until they discovered that the plants of important economy are destroyed by the fungi. So now they start to study them to see how to stop them, how to take care of those type of infection. In the Midwest, they had, few years ago, they had infection of their economy. I think it's South Dakota or Missouri, one of the two. So the the economy of that state is based on chestnut. Chestnut are the trees where you get the wood for furniture, all kind of table and so on. So it's very important in the economy of that state. So what happened is, one time there was infection with a fungus that infects all the chestnuts were. So it was a serious problem, a setback in the economy. So they had to destroy all those chestnuts. So the question was, if they plant the new one and they take years to grow, so what tells them that they will not be infected again? So they went back to the lab, to the scientists in the lab, through biotechnology, to modify the gene in chestnut to have the strain of chestnut that is resistant to the fungal infection. That's what they did. So now they have the chestnut that cannot be infected. So you have some fungi that are beneficial to us, important in the economy. We already saw the Saccharomyces cerevisiae that we use in all this, and they use it also as model of eukaryotic cells because it's well known, it's not pathogenic, has few genes, so they use it to make the vaccine to hepatitis B virus. That is one of the first vaccines. So they use Saccharomyces cerevisiae, so that the results that they get from that research, they can transfer that to human, since this is a eukaryote. You have some like entomophaga that is used for biocontrol vocabulary. What is biocontrol? It's the control of living organism by living organism. So this fungus is able to kill the mosquito, which for a long time they were using the chemical to kill. You had the truck with those fumes pumping in the street, but one street after another in the evening to kill the mosquitoes. But now the result was people who inhaled that developed the problem to their lung, the cancer, the lung problem. So now they don't use that anymore. So they found that entomophaga, the fungus, can do the job. So which is good. So in here, I will expect you to know for Saccharomyces cerevisiae and for entomophaga, you know the definition of biocontrol. On the other hand, you have the fungi that are pathogenic, that cause the diseases. Those are the disease caused by the fungi is called mycosis. The mycosis, like athletic foot is a mycosis. Okay. So now, based on what part of our body and how deep is the infection, determine all those terms here on mycosis, all those terms. Like systemic mycosis is the mycosis that we can get through inhalation or through the injection, and that will be carried through the body by the blood. So this will cause deep infection. That means all the parts of the body will be affected. So you want to take those in relation of the explanation here. Subcutaneous mycosis comes from occupation often. That comes from occupation. Like you walk in the garden and you have puncture of something, and torn or something that is infected with a type of subcutaneous infection, and that will develop beneath the skin. While cutaneous, they grow on the surface, and those surface can be hair, skin, nails. You see sometimes the nails they change color. That is fungal infection, it's mycosis. Or the nails that become flat, instead of being rounded, become flat. That also is a fungal condition. Or the nail become very hard and break easily. So all those are the cutaneous mycosis. You have what is called opportunistic mycosis. Opportunistic is about opportunism. So a microbe like Candida albicans. Candida albicans lives in vaginal tract. And Candida albicans there has no power, it stays quiet. Why? Because lactobacillus, the bacteria that lives there too, produce the lactic acid, making of vaginal tract the overall acidic environment, which is not good for Candida albicans, so it's quiet. It's kept in check. But now when you go see the physician, you have ear infection or another bacterial infection, they give you antibiotics, then those will kill not only that infection, but kill also lactobacillus. Then the Candida albicans will become happy, will cause yeast infection. Okay, so that's what we call opportunistic. That will hit when you are immunosuppressed. We will stop here. Underestimate this chapter because it looks, you have a lot of parasites. We talk about, the sleeping disease and things like that. But you will forget that it's not easy because the terminology are not something that you hear every day. Okay.